Okay, Anatomy and Physiology, good afternoon. This is Mr. McGee, and we're going to try to make another video tutorial, but this time with our Osmosis lab that we've done. And I'm going to try to make it as quick as possible so that everybody can kind of see how this is done. So if you follow very closely, you shouldn't have a problem. But again, ultimately, you're pasting your stuff on this report and answering the questions. Let's go ahead and get started. First thing, let's go ahead and take a look at this and make sure we can add our titles. This is our independent variable, and this is all our dependent variable, and we're going to want to make sure we get these titles in there. So you're going to want to add a column over here, and you're going to want another row on top. So to add a row, you just simply kind of click on the top cell and right-click, or on a MacBook, you use two fingers and hit, and it makes it right-click, and you go here to Insert Row, and then you can do the same thing as long as you're here and go to Insert Column, and that should do it there. Now we want to get our title right in these little cells, so in order to get these together merged or whatnot, I'm just going to take this amount of salt dissolved and I am going to paste it into this little cell right there, and it doesn't matter because ultimately we want it grouped with this right there. So what I'm going to do is I'm going to do this, highlight all these cells, and I'm going to go ahead and merge them together with this merge option, just like that. But it doesn't fit, so now I'm going to go to this wrap text option. That's going to get it to fit in there, and then I'm going to come here to vertical align just to kind of make it look nice. And if you want it centered, you can even go here to center and make it nice and centered like that. Okay. Likewise at the top, we want to put our change in mass in any one of these cells up there by copying it, and well, on yours you're going to have to type it in, and that's our change in mass. But we want our change in mass to be in this entire top row up there, and to do that, just like before, we need to merge the cell together, and then we need to center it just like that. So now that we've got that up there, we need to make some boxes for our table. Let's go ahead and box all of these together, and let's go ahead and hit the border sign, and let's do the same thing over here and make a border sign. Notice I don't have any border over here because that is not necessary for these areas. So now that we have our borders, let's go ahead and do something else. You can just kind of highlight all these cells here and go to center. It gets our data nice and centered. We need to get our mean. And in order to get our mean, we need to calculate the following formula. Equals average. And then you hit enter, or I think it's return or something on a MacBook. And then you're going to highlight your raw data from group 1 to group 6 over here. By the way, these are my data from both the different classes that gave it to me. Hit enter and it gives you your number. And then you just do that for each one of these here. Hit enter, and then you just kind of highlight the data and hit enter. Another thing you could do is you could just click on a cell there and you could copy it and you could paste it. That way you don't have to enter it for each one of these. It's kind of a quick little shortcut. Now that we have our table, let's go ahead and add a little top border up there just like this. And if you hold Control, or on a MacBook it's Command, you can highlight it just like that here too. And I'm going to go here and add a little background color. You want to add a background color to this. Might be a little dark. No, we'll leave it at that. You want to add a little background shade because that way we can clearly see the titles from our raw data there just like that. Okay? I will leave it to you if you want to make different colors or different border labels or border thickness, but make sure it looks roughly just like this before you copy and paste your graph. But we need to do more. Notice the decimal points. Some of these are rounded to 2 decimal points, some of these only to 1, and some of these are rounded to a whole number. That's not good. You need to highlight all of this and go up here to your decimal point options, and you need to add and remove decimal points. Now, if you think about it, we were weighing everything to the 100th of a gram, which means we need it to 2 decimal points. And a rule of thumb, you generally want your means or any processed data to be 1 more decimal point than your raw was. That's kind of a good rule of thumb right there. So go ahead and do it like that. And with that said, our table looks very nice and our table looks done. I mean, if you want, you can even bold it like this. That's kind of up to you guys. But as long as your table looks roughly like this, you will be fine. Now to go ahead and copy your table, if you're using Google Sheets, you unfortunately cannot copy it like this. It will not paste it. So we're going to need another option. If you're on a PC, there's what's called a snipping tool. And a snipping tool, you just basically do this. You crop it and then you basically just copy it like this, and you can paste it on your file like this under table. And from here we can size it. The problem is on a MacBook, which we're using in class, you cannot do that. There is no snipping tool. On a MacBook, you have to do the following. You have to do Control plus Shift plus 4. Control, Shift, and 4, and then you crop out your table, and then it automatically saves it to a picture file. You can use that picture file on your desktop to paste it as a picture. So I will let you guys decide what you want to do with that. Let's make our graph at this time and see how this works. And again, if you made your table exactly as I made mine and in the same sequence that I made mine, you should not have a problem with this. But to make our table, we need to manually tell the computer this. If you highlight it, this is our x-axis. Now you're going to hit on a PC, it's Control, or on a MacBook, you hit Command and hold Command, and then highlight the dependent variable, our y-axis. So again, you highlight both of these, but you're going to actually hold Command or Control between when you hold this and that one there. Okay? Once you have it highlighted, go up here to Insert Chart, and you're going to end up with something like this, because one thing I don't like about Google Sheets is Google tries to figure out what your graph is without even knowing it. So you're going to go up here to Column Chart. We want this to be a scatter plot. Okay? Google, for some reason, tries to figure out what the graph is before you do anything, and that's a problem. So you've kind of got to do some interpretation on your own here. But this is what our table is supposed to look like. So let's go ahead and work with this option that we were given. And now that we have that, we'll go to Customize. And what you're going to do now is you're going to modify a few things in here, such as your tread line. I go under Series, add a tread line. And you want to find an option of the tread line that fits the data best. Polynomial, do you think exponential? Do you think power series or logarithmic fits best? You pick the one that fits the line of best fit, the best, and then that's going to be the one that you're going to stick with. But the next thing we need to do is, after series, you need to add a title to your axis. We need a title for the dependent and independent variable. Go here to your horizontal axis title, and that's going to put a title at the bottom here. And under title, you're going to type in the horizontal title. It should be the same thing as over here. In this case, the amount of salt dissolved in 100 milliliters of water. You can even change your font size if you want to make it bigger. You can make it bold. You can do all that, and I'll let you play with it. But now we need a label for this axis. We're going to go here to vertical axis and add a title. Now, you would think it should be change in mass, because that's what's above. But keep in mind, it's not just change, it's actually our mean change, or our average. So I'm going to add that in there. Our mean change in mass in grams. And again, you can make this bigger, you can bold it. I will let you decide what you want that to look like, but make sure that that says average or mean change in mass. Once you get that done there, your graph for the most part looks sufficient. If you have a problem, you can always delete it and go back, but when you copy this, do not click on the middle here. It won't copy anything else. You click on the outside like this, and then you go up here to copy or hit Control-C. Go up here to copy, and then go on here on your Osmosis lab report, and then you go to paste, just like this. And you should be able to paste it on link. And again, you can center this, you can shrink it down and make it bigger. I will let you play with this. One last thing you need to do here is you need to go here and do R square again and calculate it. And in order to calculate R square, like I said, you will go back to your raw data and you will type in equals RSQ and hit enter. And then just like before, you're going to highlight your independent variable, hold Command or Control, depending on if you're using a Mac or Windows, and then you highlight your dependent variable. And then when you hit enter, it should automatically give you the results, but I'm not going to do that for you today. I want you to calculate it and see what kind of results you get. This is your lab, very similar to last time. If you followed it correctly, you should have no problem, and your lab should look nice and neat for a science lab report. This is Mr. McGee. Good luck with your labs. We'll see you later.